Hey guys, we're going to jump into the origins of Earth and kind of history of life. First set of notes here for evolution. So it's 80s day. I got my cassette tapes on. You guys like that? So we'll get in here. Here's a basically a dinosaur that's been around, I think 35 million years. That was just kind of an interesting picture in your textbook that I saw. Kind of an interesting creature. It's actually more related to a mammal than it was a reptile. And I think that really defining feature of that sail on its back is pretty interesting. Scientists think that it might be used for temperature regulation and or attracting a mate. So pretty interesting little critter there. So big ideas. How old is the Earth and what was it really like? How did the organic molecules first form, and how has our planet influenced the changes in life over time? So another picture from your book. This is actually a picture. If you can look at the layout of the bones there, I'm guessing some of you can figure out, or most of you figure out what that is just based on the size of that thing. It's actually a whale, and that picture is from the Sahara. So the Sahara Desert. They've apparently found many whales in the Sahara, meaning our planet has not always been like this, right? Like there have been lots of changes over the millions and billions of years, and so at some point that must have been underwater, and marine organisms perish there, and they find bones of things all the time. Actually, I was taken to a place at 270 and 44. There are some rock cuts right there along the highway, and if you look in the rocks at the bottom, if you actually like park on the side of the highway, which is not a great thing to do, but we were doing it actually as a field trip in college or a grad school class. We went down there, and if you sift through the rocks down there, you'll find fossils of mollusks, which means that at some point, even though we are in the middle of the country right now, at some point we were underwater as well, millions of years ago, because otherwise there would not be marine mollusk fossils there. So pretty crazy to think that St. Louis was at some point underwater. So how do we know what we know? It's kind of hard, things were millions and billions of years ago. So how do we know what happens? Different ways. We're going to go through some of the different ways. Part of it, you guys probably are more familiar with like deductive reasoning. Deductive reasoning, you kind of, well, I know A is true and I know B is true, so that must mean that, C has to happen, right? And you kind of start with these broader ideas and you deduce and you get down to some details of how things must be. Induction or inductive reasoning be the opposite. We're looking at very specific pieces of evidence, like cracks in the earth right there that you can see in the picture, and then go working backwards to like, well, how do these things go? And you come up with these much broader ideas, and eventually you work up to like plate tectonics and how the continents have shifted around. So induction is kind of the reverse of deduction type of thinking. So are dates really accurate? When you see things that, how scientists, people, teachers say how old things are, is it really accurate? Well, pretty close, yeah. Some of them are ranges, and granted, they are, some of them can be bigger ranges than others, but they're still pretty good. So one way is to use radioisotope dating. We've talked about isotopes, radioactive isotopes already this year. And what happens is when you have these radioactive isotopes and they decay, they give off particles and radiation, and they end up changing to like another substance, another element, pretty much. So like if you look over here in the chart, the radioisotope carbon-14, and your decay product is nitrogen-14. So things change over time, and when they decay, they actually become different elements, potassium to argon and rubidium to strontium and so on. And so if you look at the chart, hopefully my pen will work today. If you look at the chart over here, here's the percent isotope, okay? Here's the percent isotope, so 0, 25, 50, 75, 100. So decay of radioisotope. So here's the original. The blue line would be the original element. So we're at 100% up here, okay? And the buildup of the decay product, well, if this one's at 100, then this one has to be 0, okay? And so then we have to look at what's called a half-life, the amount of time for exactly 1/2 of the isotope to decay. And the half-life, the amount of time that takes is different depending on which radioisotope you're talking about. If you look at carbon-14, which is a popular one, you guys have probably all heard of carbon dating. Carbon-14's half-life over here in this box is 5,730 years. Okay, 5,730 years, really long time. Nothing compared to the rest of these half-lives in these boxes down here. Okay, so if you look at those, they're much longer. But carbon-14 has a really pretty short half-life, even though none of us would ever be around that long. But they can use these to kind of work backwards and create a clock. And so if you had a sample of carbon-14 and you had 100% of it, and then it comes down when it loses 50%, that's one half-life, okay, time and half-lives. So how long did it take for it to lose 50%? Well, it took this long. It took 5,730 years, okay, for it to lose half. And now, if it's at 50%, well, what's half of 50? Well, it's 25. So then the original product from 100 to 50 to 25, and then the new one is going from 0 to 25, 0 to 50 up to 75. And it will continue on the upper arc, and the other one will keep losing, and you can kind of see how that goes now. But 0 to 1 half-life, that's 5,730 years. And then from 1 to 2 is that same amount of time, 5,730. And 2 to 3 is another 5,730. Okay? And this is going to keep going on and on and on and on. And so when you find some substance, maybe it's in a rock or a fossil or something like that, they can look and see and determine how much of the radioisotope is there and how much of the new decay product is there. And they can look at those percentages and then work that clock backwards and figure out how long something had really been there. And so pretty cool way to actually do that. Based on this type of method, the universe is dated to be about 15.5 billion. And then we have Earth being about 4.5 billion based on some asteroids. So early Earth was not like it is now, right? Not very blue and green and lush and aquatic. Excuse me. Super hot due to just forming, very toxic, no oxygen, a bunch of other gases. I'm going to pause the video real quick because I got something in my throat. Hang on. Okay, much better. So early Earth, not really inhabitable, okay? So if we move on and we start looking at some of these things, so from cell theory, from like regular bio, you guys probably remember like life is made of cells and that all cells come from pre-existing cells. So if that's the case, that should, you know, kind of lead all of you to ask a question. If all life comes from pre-existing cells, what should be the question you'd be asking right now? It should be, well, where'd the first one come from? Like, there had to be a first one. So life is thought to have originated in about 4 stages. Get myself out of the way here. Maybe, maybe not. There we go. See if I can go there. So first, you need to have a few things, and it's all related to the organic molecules that we learned last unit. So there's going to be some carryover here for sure. So nucleotides and amino acids were produced prior to the existence of cells. Okay, so that's one thought. Nucleotides and amino acids became polymerized. So they got longer, they became bigger, they became more complex, and they formed DNA, RNA, and proteins. Clearly, you have to have those things if you're going to start having cells. But again, this is pre-cells. Polymers became enclosed in membranes, so now they got protection from the phospholipid bilayer. The lipids are participating now. And then polymers enclosed in membranes evolved cellular properties. So eventually, these things got enclosed and they started working together and they started doing cellular activities. Okay, so that's kind of the thoughts on some of this. So we'll go through them. Stage 1, many scientists believe nucleic acids and amino acids formed spontaneously before cells ever existed, and that would have formed some sort of primordial soup that set the stage for more production of those organic molecules. So there are 3 hypotheses that are recognized. Maybe more than one is correct, maybe none of them, but these are the 3 kind of leading hypotheses on how these things would have actually formed. We have the reducing atmosphere, the extraterrestrial, and the deep sea vent. So the reducing atmosphere, you guys might have seen this experiment in textbooks or in earlier classes. It's called the Miller-Urey experiment. And so what they did is they tried to mimic the conditions of early Earth. So they set up this apparatus here, and they tried to, like I said, mimic the conditions. They have a bunch of gases in there. They have electrical charge. Why do you think they need electrical charge in there to mimic Earth conditions? That's because there was a lot of lightning. Okay, so gases give up electrons easily. We know there was a lot of gases on the Earth back then, despite there not really being oxygen. There were a lot of other gases. And so you have the sparks from the lightning. You have the Earth is cooling, right? The Earth was very warm, and now it's cooling over a very long period of time, but it is cooling. So that's why you have the condenser on the right side of the image there, so that you're cooling things off, and then you have the trap there to collect anything that might form based on this rearrangement of electrons in these conditions. And so detractors do think they have the wrong atmospheric conditions, but this is just one picture. They used many different setups, used many different conditions, and just about all the time molecules were produced in some way. So when they looked in this flask when they got done, there were proteins in there, there were nucleic acids in there, amino acids. So there were organic molecules in here. So this is pretty strong evidence that molecules could have formed this way back then. So another way, some scientists believe that the molecules would have, better formed by volcanic or sea vent, and so they tried to recreate those conditions, and they actually got more. And Miller was involved in this as well. So they actually got more amino acids, doing it through a kind of volcanic setup, mimicking the volcanic setup, than they did the reducing atmosphere way. So either way, it's showing that amino acids and other molecules could be produced kind of without cells, just kind of on their own based on the conditions that early Earth were in. And so you see, like in 1953 with the Miller-Urey experiment, the number of amino acids and the mass of amino acids produced. And then in 2008, clearly better technology, and they're able to identify more things. I think Miller-Urey in 1953, I read once that there were actually more amino acids there than they found, just because they weren't exactly sure what they were looking for and didn't have the technology to do it. But anyway, a couple good ways here that molecules may have been produced. Then we'll kind of tackle the other two here on the one slide. Extraterrestrial, right? When you think of extraterrestrial, at least when I think of it, it goes back to the old, again, 80s movie for our theme here at school today, E.T., right? E.T. was the extraterrestrial, right? So not of this land, not of this earth. So that means we're talking about meteorites. So meteorites contain tons of carbon, and they also have amino acids and nucleic acids, just molecules. And so could meteorites have carried enough organic material to Earth to kind of stock the soup? I'm sure, around the days of the Big Bang, things were flying around, and I know Earth got bombarded quite a bit. So would there have been enough, carbon and molecules to, kind of stock the soup for then those molecules to take off from there and form cells, etc. So who knows, but it's a hypothesis. And then the deep sea vent is another one. Conditions suitable for organic chemistry. If you go, there are some deep sea vents, and a lot of proton gradients happen down there in that organic chemistry in the sea vents. And so proton gradients are kind of how cells power themselves. So that energy side of things has a little bit of bearing on this hypothesis, and how you can't just make the cells, you have to be able to power them. And so maybe this is one way that things would have not only been able to be generated, but would have been able to power themselves. So just another hypothesis there for you. Stage 2, polymerization. Let's assume we got some molecules built. How do we make them bigger? How do we make them more complex? How do they join? So organic molecules alone are not enough. So They have to be able to polymerize, carry out more advanced functions. It would have been really difficult in aqueous solutions. Why? We'll come back to that in 1 second. Let's look at this graph. What do you notice about the graph? Take a minute to pause the video if you want. And when you're looking at this, it's like the number of molecules that are being formed here, and it says precursor molecules only on the red line down there. And then on the blue line it says precursor molecules plus the Montmorillonite clay, and way more, right? You get way more molecules. So it looks like it would have been better to form them in some sort of clay, to have these molecules polymerize and get much bigger in some sort of in clay or on land versus water. And back to that question, if you remember, well, what reactions build molecules and what reactions break them down? Dehydration synthesis builds them. What reaction breaks them down? Hydrolysis. What do you need for that? Water. So maybe that wouldn't have been a great area for polymerization to be happening, because hydrolysis would have been more likely to occur there and would have broke things down, not built them up. Stage 3, primitive cells form. Protobiont is what they call the things before cells, the first non-living structures that evolved into living cells. So they had some things very similar to cells, and they were almost there, but we just don't call them cells. We call them protobionts. So 4 things they had. They had a boundary, okay? So they had a membrane-ish thing. They contained info, which meant nucleic acids. They were enzymatic, meaning they could do things like proteins, and they could replicate. They could copy themselves. So those are all kind of the main characteristics you need from something in order to have them take off and then evolve cellular properties. And then 4, we get into something called the RNA world. The RNA world hypothesis is that RNA was probably the first information molecule, not DNA. And there's a couple reasons for that. RNA can store information just like DNA. It's made of the same bases, so it can store information too. But it can replicate, as can DNA, but it can replicate, it can copy itself. And then the one that DNA doesn't really do that RNA does is it has enzymatic function. RNA can do things. DNA really doesn't. And we have these things called ribozymes, and they're kind of a combination of RNA and protein. And they actually do things. Now, when I say do things, we're talking about really simple things. And so let's look at some simple things over here, okay? So this GUN up here, this is just to indicate a little sequence, and it's to show you that, there's a strand or a molecule there. And then we have, this is actually part of the RNA. And I know you've heard me talk that there really is double-stranded RNA, like that's a thing. And it happens in pieces, it's not always, but this sort of structure right here, okay, this is called a hairpin loop, and you don't need to know that yet. We'll get into this more in genetics, okay? But yeah, I wanted that drawing to go away. And then right here is kind of where it doubles back on itself, those bonds there, that's where double-stranded RNA is forming. But I'm getting ahead of myself here. So if you look here, this molecule is with the plus there, they're going to attach. So over here are the same 2 things right here, and now they are attached and they're bonded together, right? So they've attached based on the sequencing. Let me move my face off of this picture. And then down here, this is where the actual enzymatic activity comes, and this gets cut right here. That little picture there with scissors, and they're cutting that strand from 1 molecule strand into now 2 strands. Now, that doesn't seem very impressive. It's just a little thing that shows it can do something, but that is what enzymatic means, and that's what the types of things RNA can do. And maybe that's not a big deal, or maybe it is. You can things will then change. Now you have 2 separate things instead of 1, and maybe those sequences will match with something else now, and something else will happen differently down the road. So we have what's called chemical selection. And so, grab my pen again. So really what's going on here, if you look in here, so all these yellow strands in the protobionts are RNA. Okay, so we have RNA all in here. And these are protobionts with no enzymatic functions. All the ones with yellow do not have enzymatic functions. But look what happens. We get a mutation. Okay, and since in copying itself, there's a mutation. And now we have this purple guy right here. And he's different. Okay, and he now has the ability to self-replicate. Okay, so he can copy himself. So now all of these things kind of turn from yellow, and over time they all become these purple things, and they can all do this. Okay, so now they all have a function. Okay, so it's called chemical selection because this is basically natural selection with chemicals. Okay, and so if you remember your basic natural selection, when you get some sort of change, right, and then the things that come out better or survive better will move on and advance and copy themselves. Well, this one does. And so if you look over here again, we'll look in these protobionts over here. They're all purple. They're all purple. Oh, we got a second mutation. So here we have another mutation. Now we have a green one. And this mutant RNA has the ability to synthesize ribonucleotides. Okay? So now we have a protobiont that can do two things. Okay? And it's starting to gain functions where it can replicate itself and it can build molecules. And so now they're already getting more complex and they're able to do two things from somewhere where you couldn't do anything. So you can see how over huge amounts of time, millions of years, that this would lend itself to start building something that resembles a little bit more of cells in life. Okay, so get out of there. So here's kind of the geologic time scale, and I know it's really small, but if you squint, you can see it. Actually, I can see it pretty good. So nothing you really need to memorize. I do think it's important to know a few things. The eras, this column here, the era. One second. Sorry about that. I had a big sneeze. So the eras up here, as I was saying, I would want you to kind of at least know what these are. So I would rather start from the origin. So down here is the origin of Earth. Okay, so way down in this area is the origin of Earth, and so there's actually an era listed way down here, down below. There's one called Precambrian. Okay, Precambrian is the era down there, and it's the longest one. It's the biggest one. It's from when Earth started all the way up until quite a while ago still. 4.6 billion years ago, I guess, would be the origin of Earth. And then, so this section right here would be Precambrian, and it doesn't show it that big because they had to do like one of those cuts in the graph, but far more time took place here. And then you have, the next one was the Paleozoic. Okay, so Paleozoic, when you're getting, your first cells and your first small organisms, they're coming out, some plants, fungi, different bacteria, stuff like that. Is what's going on. Starting to get into some reptiles right at the end of it. Starting to get into some reptiles. And then the next one would be Mesozoic. And this one is where Mesozoic is the age of the dinosaurs. You can see a dinosaur over here. And notice that one of the periods is the Jurassic period, where they got the name for the movie. So they have the Triassic, the Jurassic, and the Cretaceous. So organisms are getting more and more advanced. Flowering plants, you're getting angiosperms, lots more diversity. And then finally the Cenozoic. And we actually live in the Cenozoic era. That's still happening. Okay, that's from like 66 million years ago to now. Okay, so our era is 66 million years long and counting. Okay, the Mesozoic was from 252 million years ago and 66 million years ago. Okay, Paleozoic, you can see the dates here and how long they are. And we'll talk about why the eras change here in a second, but just kind of wanted to point some stuff out to you, but nothing you really need to like memorize, but just kind of be aware of the eras and kind of how things evolved, but you don't need to like memorize the chart or anything. So the first cells were most likely anaerobic heterotrophs. So break that term down. Anaerobic means no oxygen, without oxygen. Okay, and then heterotroph, if you think back, well, what kind of things do heterotrophs do? They have to go out and get their own food. They don't make it. Okay, so organisms that didn't really deal with oxygen and had to get their own food. Okay, so that was probably the first cells because the planet didn't have oxygen. So anything that was living was anaerobic and had to acquire food somehow. Okay. Photosynthetic cells developed about 500 million years after the first cells. Cells were around for about 500 million years before photosynthesis ever began. Cyanobacteria have shown to be some among the first photosynthetic cells. And that's kind of cyano. That's that you might see that on like your printer cartridges. That's that blue-green color. So that's like blue-green bacteria or blue-green algae. Cyanobacteria form stromatolites. That's what you see down in the pictures down below. And so then they can go down and date them and figure out how things might have worked back then. And then just a question for you here, or a thought. Give 2 reasons why photosynthetic cells change the world. So now that cells are photosynthesizing, how would that really change things? See if you can pause the video and come up with something there. There's a couple of big things that would happen. So mainly one of those things would be that photosynthesis led to the oxygenation of the planet. Okay, so photosynthesis takes in carbon dioxide and puts out oxygen. And without many things on the planet, like people or lots of plants or anything, the oxygen would start hanging around. It would start oxygenating the atmosphere. And so now you have anaerobic organisms. So the ones that don't like oxygen now are getting forced into like the soil. Okay, they're going into the soil to get away from the atmosphere, to get away from the environment, and they're going to go live in the soil. And then you have aerobic respiration becomes possible, and that's way more energetic. Okay, aerobic respiration versus anaerobic is way more energetic. You can make a lot more ATP. Now you have a lot more energy, and now cells can do more things. And so that's a really big deal for advancing life on the planet. And this just kind of shows you, the atmosphere goes 2 levels over time and billions of years ago, and how oxygen really took off. But look at that vertical jump right there, just before. Looks like 2 billion years ago, between 2 and 3 here. Look at this vertical jump. Well, that happened, really quickly in the, course of geologic time. Anyway, it happened really quickly, and so that really changed the game. So, and really, I didn't mention the other thing, but photosynthesis, oxygenation of the planet is one thing about photosynthetic organisms, and then just think like. Now that kind of changes how things can get food, right? Because now you have heterotrophs, but now you also have autotrophs. You have autotrophs making their own food through photosynthesis. And so now you start developing what's at the bottom of every food chain, autotrophs, right? Things that make their own food. And so now you start having these more complex food chains and food webs that are able to be formed because of photosynthesis, okay? And you have this exchange with respiration and photosynthesis, which is really what drives the whole planet anyway right now. It still does today, okay? So evolution of eukaryotes, now moving up into more complex cells. So initial cells would have been prokaryotes, right? They would have not had a nucleus. They wouldn't have been very complex. So eukaryotes are the more complex cells, and they can do a lot more things. They have a nucleus. They have all these organelles, and organelles are really just compartments, right? So what's great about compartments? Well, you can do multiple things in multiple different environments within a cell, okay? So, you have your organs. Does your stomach operate in the same environment that your lungs do? Not at all, right? We have different functions going on inside our bodies, in our different organs that rely on these different environments to get things done. And organelles are the same way for a cell. They have different environments, but they can multitask in different environments all at once with these compartments, with these organelles. So how did we get here? Um, how did we get eukaryotes from prokaryotes? So, oh, let me go back. Just trying to grab my pen. So I know it's really small writing, but again, if you kind of, you can probably blow it up on your computer. If you look at it real close, I think you can see it. But they look like super similar diagrams. It's just if you look way down here, and actually my pen doesn't even write that low, but look below that red mark and look below this red mark, way down there where it says A and B. And A and B, A says the origin of symbiotic relationship, and B says origin from endosymbiotic relationship. And that's why I put those two terms here, symbiotic and endosymbiotic. So how did it really happen? Was it one? Was it both? And what are the differences? Well, if you look at symbiotic here on the left, you basically have two prokaryotes. Okay, you have one here and you have one here, and they merged, and now you have one thing, and they're all together, and they're in the same area. And now, down here, a little further down, it forms a nucleus, right? And then the cells start taking off, and they get organelles, and they kind of go from there. And we'll talk about the bottom half of that in a minute. Over here, this is called endosymbiotic. And rather than just merging and this all being one big open area, this one over here basically gets engulfed by this one. So the red one, will say, engulfed the blue one. But he didn't merge right away. He kept his barrier there, right? So he keeps his barrier and he becomes something different. That becomes a different organelle. And you can't really tell, but there'll be some pictures here in a minute. But this is going to be what becomes the mitochondria. Okay, so if we move on a little bit here, we have endosymbiosis, which is the theory that mitochondria and chloroplasts were once free-living bacteria, but were engulfed by larger bacteria. So I'm going to put my head up here now that we've seen those words, because we're going to put some more up here. So there's a space. So evidence of this. How do we know this? Mitochondria and chloroplasts both have DNA. Okay, no other organelles have DNA. Okay, there's not just DNA in your nucleus. Mitochondria have DNA. Okay, now they are different DNA. They are not going to be able to run the entire cell, but they have DNA and they make proteins, as do chloroplasts. Mitochondria and chloroplasts reproduce independently from cell production, just because when a cell replicates, you divide up the organelles into both sides, and then you can make the others. But even if a cell isn't replicating, if a cell isn't copying itself, you can make more mitochondria. A cross-country runner at your school has more mitochondria, than people that aren't, probably. Maybe swimmers would be in that category too. People that do a lot of aerobic exercise are going to have a lot more mitochondria. And the mitochondria can replicate itself to keep up with the demand. Okay, not all organelles can do that. They have their own ribosomes, which means they're making their own proteins, and they have a double membrane. Okay, they have a double membrane, not just one membrane around the organelle, but they have two, because they had one, and then when they were engulfed by the other one, another one went around it. So it had its own membrane, and then the lipid layer formed around it again, and ended up with two membranes. Okay, and everything benefited from this, right? So if you look down at the picture now, all these cells that couldn't really do much, they end up taking in these other bacteria that were free-living bacteria, and they take them in. Well, both of these things are like energy-giving organelles now, and so some of them only ended up with a mitochondria, which means that would have started your animal lineage of cells, and then some of them ended up with the mitochondria and the chloroplast, which would have started your plant lineage and plant cells from there. So everything would have benefited in this case, because the free-living bacteria would have now gotten protection and materials, and then they would have given energy back to these things that engulfed them. So that's what the endosymbiotic theory is. Multicellularity now. So now we're getting bigger combinations of, you need me? Bigger combinations of cells. So sorry, somebody was at my door. Did multicellularity come about aggregation or division and adhesion? No one really knows. So did they clump up together or did they just divide and then stick to one another? Like, how did this work? No one really knows. How does it happen today? It's really division and adhesion today, is what people have found out. So, three principles of multicellularity. Organisms arise from single cell that produces daughter cells, okay, that adhere to one another. Daughter cells can differentiate, and as an organism gets larger, there are more somatic cells to carry out functions of the organism. This is basically mitosis, right? So there's a lot of mitosis and a little bit of meiosis in there, which we'll get to later. We won't do details now. Somatic cell, that word somatic, if you don't remember it from regular bio, are just body cells, regular body cells versus your sex cells. Okay, so somatic cells are everything else except the sex cells. Okay. So here's kind of a timeline. Okay, first multi-celled organisms 1.5 billion years ago. First invertebrates 590 million years ago. Then we have this thing called the Cambrian explosion. Okay, 533 to 525 million years ago. Okay, well, why did this happen? So, first of all, explosion, only because not like bomb, not a bomb going off. This is an explosion of life. Okay, so look at that time frame. There's only 8 million years there. Okay, now 8 million years seems crazy long to us, but when we're talking about the geologic time scale, 8 million years is really not that long. When we're talking into hundreds of millions and billions of years, 8 million is super fast for a lot of things to happen. Cambrian explosion is when we got this big diversity of life that showed up kind of all at once. And a lot of that had to do with just the conditions that were surrounding everything, where you had the oxygenation of the planet, you had these food webs and food chains forming, and there was plenty of food, and the balance between predator and prey was good, and you had things spreading out around the planet, and it was conditions were just right for a lot of that to happen. So you have first vertebrates and land plants 520 million years ago, first seed plants and insects, and I don't need to read all these. You guys can kind of look at them. But then you have your first Homo sapiens, your first humans showing up about 130,000 years ago. Compared to everything else, we are a very young species. Okay, I know you guys talk about ancient civilizations in your history classes. We're not ancient. Okay, not compared to everything else. So we're still the new kids on the block compared to all of this that's happened. And so just gives you an idea of the time frame. And again, I'm not going to ask you to memorize dates and stuff like that, but just kind of interesting to see the timeline, how things progressed. So patterns in evolution. As time goes by, evolutionary complexity increases. Why would that be? Why would evolutionary complexity increase as time goes by? Well, if you kind of look at the graph, it's pretty. You don't even really need a graph to kind of understand that statement. But as time goes by, as millions of years go by, and you think about, well, how would evolution increase? Well, in natural selection, how does that work? Well, it's the genetics, right? It's the genes, it's the bases, it's the DNA, it's the mutations, right? Over time, we collect a whole lot more mutations in our gene pools over time, and we have more mates and more things to interact with. There's more populations of all the different living things on the planet, and so the longer they live, the more interactions they are, the more mating that happens, which causes a natural mixing of DNA, which gives you different combinations, and you have mutations. And so things get more and more complex as you go over time, especially millions of years, right? That's what we have to keep reminding ourselves, is the time frame we're talking about here. So unfortunately, this is one of the craziest numbers, I think, all year. It always blows my mind away when I see this. 99% of things that have ever lived on this planet are gone. So think about all the diversity of life we have on this planet, and all the crazy things in the ocean and the rainforest, and all the small, tiny, microscopic things, and the really big things, and all the things we have living on the planet right now. And that's about 1% of the things that have ever been on this planet. So that's just so crazy to me to think about. So what happens are extinctions. So move myself out of the way here. And if you look, there have been about 5 mass extinctions over time. and it says, can you spot them on the graph? Well, not too hard to spot. So let me grab this here. And if you look, you have total extinction rate going up. But look at how, life kind of goes along, and then we're doing well, and then we crash. So, here's an extinction. 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 Whenever you see that big line drop, okay, so on the right-hand side, it's number. You have number of families, and so you're looking at when those things really drop. You have these mass extinctions, and so that's really how the eras are divided. Anytime you got to the Mesozoic era ended because there was mass extinction of the dinosaurs and a whole lot of life on the planet. And so why do we get these mass extinctions? Lots of reasons. You can see hopefully over here. Whoops, let me go back. I know my numbers are covered up. There it is. Hopefully they go away. Lots of debates. Climate and temperature. We might be heading into one of those now. We definitely have an issue with our climate right now and how things are going to go in the future. We're certainly not helping ourselves. I'll put it that way. And that is a entirely different discussion. But that's going to be a problem down the road, and it already has been for a lot of organisms. The atmosphere, the gas composition, over time may have changed and made it hard on organisms. Continental drift definitely made it hard on organisms because you're drifting into different climates, colder, warmer, things that can't survive. Floods, glaciation, glaciers, so ice ages, volcanic eruptions, meteorite impacts. There's several pieces of evidence around the planet about giant craters. I know there's one in Mexico. I forget how big, but enormous, where the meteorite that must have hit the Earth caused so much dust to go in the air that it blocked out the sun for years. And so, there's lots of different ways which ones did which ones. You can go back and look up. We don't need to necessarily focus on that right now, but if you're interested in that, it'd probably be pretty cool to look up. Some more patterns in evolution here. Radiations. So radiations are basically the opposite of an extinction. So rather than having things all die out at once, you're talking about a lot of different things come to life at once. And so when the number and diversity of an organism goes up rapidly, and you can see this over here in this chart, where you have that little point, and then you have everything else kind of radiating out. And it looks like all the tree lines are, they're going to the right, but all these different families of things are showing up in what a relatively short amount of time, all from that one point. Why does that happen? Why do radiations happen? It's because everything's going great. It's because the climate's good, the food webs and food chains are balanced. You have lots of mates to pick from. Populations are going crazy, and then you have just normal evolution and natural selection taking place, and you have changes coming over time. All the organisms fill the niches that are able to be filled. I guess my picture's over the finches up here, Darwin's finches, and that one little archipelago of islands there, I think there were 14 different types of finches. That's, kind of weird actually, but when you go back and you look at natural selection and how it goes, that's what happens is they adjust to fill every single little different niche so they can compete in their own little group versus against everybody, because then it's harder if you have to compete against everybody than just against your little group. So anyway, adaptations lend themselves over time to that. And islands, I didn't mention islands, but other than the Galapagos, a lot of radiations happen on islands, things that haven't been inhabited before, because there might not be predators there. And as long as there's food supply and you have everything you need, there's no reason that life wouldn't just take off in an uninhabited area. So islands are a pretty good spot for radiations. Keep moving my head out of the way on each slide here. So human evolution, not really a great record. This graph is a little sad. So the Holocene is the time where humans have been on the planet. So, and been able to interact and move around. So, have an impact on the world. But the last 11,000 years, humans have been able to move around and cause and impact the planet in some way, right? Um, their force has been noticed in nature, right? About the last 11,000 years. And so if you look at this graph and you look at the total number of extinct species against the human population growth, they mirror each other. So the red line and the green line are pretty much the same. And so as we've gotten more and more humans, we have more and more extinctions. I wonder why that is. So we haven't always done a good job of protecting our species, and we're not doing too great a job now. I mean, it's, years ago when I was a kid, it was, of course we're going to have polar bears. Now polar bears might be gone by the time, in your lifetime, and definitely in your kid's lifetime, which just seems unfathomable to me that we might lose polar bears. But just can't get all the people on the planet moving in the same direction to solve this problem. So we're going to end up losing some things, like our buddy down here in this corner. I'm not sure how many of you would know what that is, but that's called a Tasmanian tiger. And Tasmanian tigers were actually in Australia, and Australia was settled by European people. People from Europe moved down to Australia and colonized it. And through overhunting and taking up their habitat and introducing dogs and disease and stuff like that, we no longer have Tasmanian tigers. So it's kind of going that way for a lot of species right now that are struggling. So I hope we can get our act together as a people, as a world, but right now it still looks like an uphill battle. And that's for a lot of different reasons, I suppose. Here are some practice questions for you. Make sure you can handle this type of information. I hope this was helpful and not too long, but I'm glad you tuned in. And I can't get my cursor to hit the button. But anyway, if you have questions, let me know. But hopefully that was helpful for you. Thanks.